Solar for special setup for me. Ooh,
All good?
good.
All right.
Hello, everybody. I want to talk about communication. And in terms of communication, there are various ways. I can use my gesture. I can touch you if we are very close to each other. You can listen to me. So in terms of communications, there are various ways. What about cells? How do you study cell-cell communication? Because you are basically computational biologist, what I know is that there are ways to study cells and communications in single cell and spatial biology. In single cell biology, we use ligand receptor co-expression. One of the reasons is that that's the only way that we can rely on. But somehow it's a little bit awkward in that we study communication after separating them. And what I wanted to say is that cells use various ways of communications. They don't touch, however, they feel what is next to them. They can sense the stiffness of the neighboring cell. They can sense the pressure on it. They can sense some other things. And there are some other mechanisms, such as gap junction, that cannot be dictated by ligand receptor co-expression. But still, we rely on ligand receptor co-expression when we study cells interaction, even in spatial transdermics. So we started to thinking about this problem when spatial transdermics is not available, and we thought about, okay, what about using doublet? Or people call it physically interacting cells. The idea is that, is there any specific genes particularly observed in the doublet or physically interacting cells here, but not expressed in single cells or homotypic doublet? To make the long story short, we performed the experiment, obtained the doublet, and found that there are specific markers such as LHX5, GSC, which is particularly observed in the doublet of neuroprogenitor definite endoderm, but those are not observed in the individual cell type. So there are something, and I want to stress that it has nothing to do with ligand receptor. We validated using various approaches, but I just want to make it short. Okay, now this is the time for spatial transdermics. You can regard one hexagon as one cell because there are technology to perform it. And now we have many doublet. I have doublet here, doublet here, doublet here. But instead of using doublet, I thought about, oh, what about using the two-dimensional space and identify those blue cells surrounded by at least one yellow cells. That can be your foreground. And blue cells surrounded by blue cells can be your background. So this is very similar to the case of doublet. And we performed it, and we found that, okay, this is a case of this data, and we found that there are some genes like PITX1 is highly expressed for gut tube cells, only gut tube cells. These are all gut tube cells, only sitting next to neural crest cells. So the red circle is for the gut tube cells, which is just next to neural crest, and we visualize the expression of PITX1 there. Okay, we identify those. What about next generation based sequencing based spatial transdermics data? In this case, SLICE, which has 10 micrometer resolution. With that resolution, you can have only one single or two cell type. It's possible to have three cell type, but not a lot. So basically, we have the blue cells and the, transcriptome composed of the blue cells, transcriptome composed of the yellow cells. We call it homotypic base. And also we have the mixture of blue cell transcriptome and yellow cell transcriptomes. It's the same case of the doublet. So now this becomes your foreground, and the blue cells and the blue and yellow homotypic pairs can be your background. And what is the expression particularly observed in this heterotypic pairs compared to these homotypic pairs? Then you can identify the genes induced by cell contact or cell interactions. So we found it, just one example here. So these are tumor regions, R1 and R2. And we have identified heterotypic pairs of the monocytes and tumors, and those heterotypic pairs expressed F13A1, and we try to visualize it in slide 6 assay. And even though these are two different tumor regions, F13A1 is observed in this heterotypic pairs of two cell type, and interestingly, they are observed in these, not in the periphery, but in the central region of the tumor region. So there are ways to study cell-cell communication induced genes by looking at spatial transcriptomics data. Then the question is it from A cell type or B cell type? So this is one example. So VTI1A is expressed in the heterotypic pairs of the monocytes and hepatocyte. If the hepatocyte portion is decreased, then this niche-specific gene expression decreased. The conclusion is that VTI1A is expressed in monocyte when they are sitting next to hepatocyte. So if we use regression model, we can predict the source cell type. That's the message. So anyway, I just wanted to compare the prediction using our approaches and the approaches relying on ligand receptor, which is NISNet, and there are little overlap. One message is that the gene list we identified is different from ligand receptor mediated genes. And second message is that I don't trust it a lot. Okay, what about Visium? That is it's the unpublished result. In Visium, it's a little bit tricky because there are a mixture of many cell types, as you've seen here. Then how can we detect the genes induced by cell-cell contact? It's a little bit tricky because we do not know the location of cells, but somehow we can find it. So one of that is we can somehow calculate the expected value, because after deconvolution, we can have the cell type portion and we have the gene expression. By multiplying it, we can calculate the expected value, and we have the observed value, which is just the Visium expression value itself. We just compare that. And then we have a list of the genes which is potentially induced by cell-cell communication. Then we used a regression model. It's a little bit complicated than this, but we applied regression model with, to identify the cell type that express the source of the disease-specific genes. Anyway, so this is just an example. So we just use the public data set because we use this data set because we have single-cell analytic, Visium, and Cosmix data together. Then these are the ligand receptor, the list of the ligand gene related with PIGR, and among them we applied CellNeighborEx version number 2. We recapitulated most of them, not all, but we are not sure if they are all true. But anyway, so our approach can recapitulate the ligand receptor genes. At the same time, we try to see if we can identify the, it's a bit slow, identify contact-specific gene. However, it's a little bit tricky to get it. So we try to come up with a strategy to make the pseudo Visium data from slide exact data by merging consecutive spots together. So it's about 60 micrometer resolution and applied it applies CellNeighbor version X2, and this is the CellNeighborEx version, which applied to slice-seq data only, and we identified pretty good overlap. So even using Visium data, we can identify cell context-specific gene expression in a pretty good way. So this is the result that I introduced in slice-seq data, and we applied the CellNeighborEx version number 2. This is F13A1 that we identified to be neighbor-specific, and we have seen that this is show up after applying CellNeighborEx version number 2. Okay. Just another story. Let's assume that we have many spatial transformatics and how to combine them together. So anyway, to make the long story short, because I don't have much time, we think that neighboring cell information is very important in matching spatial transformatics data. So we try to match the reference with the query. So it's basically matching query to the reference. Together with the transcript of the bead and the neighboring cell information, then it really worked well compared to other approaches. And these kind of approaches can be applied to map Visium H data to Visium data, or vice versa, or sick fish data is also fine. Anyway, so let me conclude with the summary of this one. So Vesalius version number 2, which is mapping spatial transformatics data, is in bioRxiv already. CellNeighbor version 2 is actually we are now writing, so it's not available yet. And I just wanted to stress that there are many ways to study cell-cell communication, and this is one of the ways to perform it. Thanks a lot.
You mentioned here, I think, Visium. I know it's a different tool, but like, how would you use multimodal data in this type of system?
For CellNeighborEx, we don't use multimodal data because we want to detect these gene expression changes. However, Vesalius version number 2, which is matching two spatial transformatics together, we can use multimodal data because inside it, we just use the color code system, basically. So we convert all the expression level into the color code. And also we can convert other modality into color code. We can just combine them together and just perform PCA together. So it's possible.
The reason I'm asking, it's I think, easier to combine expression profiles of different cells, but like the epigenomics profiles and how you combine them, if it's from different cells, is a little bit trickier, I would imagine, right?
If at the moment it's difficult for CellNeighborEx, but when it's not easy to combine multimodality at the moment. Yeah.
Yeah, quick question.
So do you infer CCI between spots or like cell types?
Say it loud, please.
Do you infer the CCI between spots or cell types?
CCI between spots.
Between spots or cell
Inside
types?
the spot, I may say.
Mm-hmm.
If you look at inside, there are cell-cell communication there.
Yes,
then
sir.
we try to detect it. If spot is large, if spot is small, then we try to detect the interaction across the spot.
Okay, so individual cell-to-cell
Yeah,
communication.
individual cell-to-cell communication at cellular resolution is possible.